So I think a lot of the kind of concerns that we're expressing here about what happens in a cell have just been sort of brushed aside on the basis that that cell will be destroyed. So if the cell is going to express a foreign protein, it will be destroyed. So, and that's true. It's true. And that's problematic, when it's just gone into that endothelial cell. If you destroy endothelial cells because they're expressing spike protein, then you create all the things you need for an environment where a clot will form. And if it's a tiny, tiny one endothelial cell, that will recover. But if you have a patch of endothelial cells, the recovery, like a skin wound, comes from the outside in. And so if it might take longer, and then you might get a more serious clot forming. But when the thing is that there are circumstances, I think, where some of those cells haven't been destroyed. And that's why we've ended up with people who are still producing, where you can still find vaccine mRNA in their bodies now.
Yeah. Something just occurred to me. If, and we know it's not true, but if it were the case that the mRNA vaccine just remained in the deltoid, we know it's not the case. But if it were, then the RNA would go into the deltoid muscle cells. The deltoid muscle cells would express that foreign protein. Those deltoid muscle cells would then be attacked by the body's own immune system. That's going to make your arm weaker, isn't it?
Yeah. So
if you're killing muscle cells.
Yeah, I think Mark did a kind of sum with me on this when we were saying, if all of those 6 trillion went into one cell each in your arm, how much arm do you lose?
Pretty well all of it.
You lose a litre of arm, we figured out in the end.
Yeah.
I mean, obviously that's not what happens. And any one cell will have multiple going because, they're grouped together. But we were just sort of playing with the maths of it.
Playing with the numbers. I must say I'm not as good at press-ups as I was, but there could be other factors involved in that. Yeah, so here we see a lipid nanoparticle going along a small vessel, and then it just goes into the endothelium of the blood vessel. And it's just disappearing inside. So when we're thinking about the development of blood clots, Claire, is the condition and quality of the endothelium, the lining of the blood vessels, an important factor in keeping blood liquid or in facilitating the start of a blood clot?
Both. So the endothelial cell produces like little anticoagulants that just say, keep everything flowing and smooth and can't make it so it can't clot. But also in order to have a clot, you need to have some sort of nidus for that clotting to begin on. So the absence of an endothelial cell creates that. It's a wound that needs a clot. So you've got both. You lose your anticoagulation and you produce a wound.
So it's a double whammy. You lose the anticoagulant effect of the healthy endothelium, and then that damaged endothelium itself becomes potentially thrombogenic.
That's it.
Yeah. This is Vircho's, isn't it? Vircho's triad, from what I remember.
Yeah, Vircho's triad has got the. I think you're going to test me now. Has got the.
It's the vascular endothelium, the viscosity of the blood itself, and the turbulence of the blood flow.
The flow, yeah, yeah.
Yeah, neither of which we want, of course. Immune evasion. So this lipid nanoparticle here is containing the mRNA, and that means, to my mind, that the body's immune system can't get to this mRNA. Because we've got foreign mRNA, normally the body would destroy that.
Yeah, well, so the mRNA, foreign mRNA and DNA naked is highly immunogenic, like dangerously immunogenic. It would cause an anaphylactic response in people that don't normally have.
Really?
So yeah. So that's one of, obviously they are using the lipid nanoparticle to deliver it into a cell, but also to protect it from the immune system.
Yeah. So really, we're conning the body's immune system here to ignore something which is normally very, very immunogenic.
Yeah, we are.
It's all just hidden.
Yeah.
Yeah.
Whereas if it was a virus particle, when that's in the blood, it looks foreign to the immune system.
Yes.
Whereas a lipid nanoparticle has no protein on the surface that looks foreign. It's just lipids. So it can get it bypasses all the sort of protective things that we have. And of course, being an injection, it's already bypassed multiple layers.
Yes.
We're breathing in things all the time, and we have all sorts of layers of protection for what we breathe in.
Yep.
But if you inject, then it's in regardless.
Yep. And also, normally, if there's superficial bacteria, that they'll be taken up by the lymphatic system and go to the lymph nodes. But if you're injecting, you're bypassing the body's natural lymphatic defences as well.
Well, yeah, I mean, the point about the deltoid injection was to, the sales pitch was that it would get into those phagocytosing cells that are picking up debris that live in the muscle, and they would go via the lymphatics to the lymph node and educate the immune system in the lymph node. So that was the sales pitch. Was not that the public were told that, mind you. That's what they were telling the doctors, was it was going to get to the lymph nodes. The public were told it would stay in the arm.
Yep. And this also is very important for the DNA contamination, because we now know that the vaccines contain DNA contamination. And I believe you're going to correct me on an error I made shortly on this, but we know that there's DNA contamination. And the levels for DNA contamination that are allowed inside vaccines were actually created, those levels were created assuming that it was a naked DNA that was being injected.
That's right, yeah.
Which
and then
we quickly got rid of.
As you've said before.
Yeah. But now we've got the DNA, foreign DNA, that is protected by these lipid nanoparticles, so it can go straight to our body cells where it can potentially do damage.
Yeah, and one thing we haven't mentioned is that when they were sort of trying to get permission to do this, they had done animal work showing where in the body these lipid nanoparticles went. And as part of that work, there's a submission to the Australian regulator, which included images where they had done immunofluorescence of individual cells to show where it was going within a cell, as well as which organs it reached. And you can see it in the Golgi apparatus. It's lit up green on the immunofluorescence next to the nucleus, but you can also see some in the nucleus. So the lipid nanoparticles were entering the nucleus, and that again, they are designed to. That's kind of what the sort of primary design of lipid nanoparticles was. Now, to be fair, that primary design has evolved into many different types of lipid nanoparticles, so you can kind of pick your tailor-made one that has propensity to reach different parts of the body and different parts of a cell. And these ones are not ones that are designed to go into the nucleus, but that image showed that nevertheless the nucleus wasn't entirely immune from them.
So the lipid nanoparticle itself, or components from the lipid nanoparticle, were getting into the very heart of the cell, into the nucleus of the cell. Yeah,
and again, actually that doesn't matter if the cell gets destroyed.
Right.
Doesn't matter, does it? It's dead.
Right.
It matters only if the cell is somehow evading destruction. And that I think there are parts of the body where these lipid nanoparticles can get to more easily than immune cells do. So in the testis and the brain, you've got these protected areas where we don't let the immune system come and attack, because we don't want to. The brain's got to be protected, and testis is only the cells in there are producing half of your DNA, so they have a sort of more foreign look to them. You don't want them attacked, but the lipid nanoparticles could get in there.
Yeah, but they wouldn't be attacked by the full force of the immune system, because those areas have to be protected. Yeah, makes perfect sense.
Yeah, I mean, we say that, but of course, in some people they were. There is a definite signal in the vaccine adverse reaction databases for testicular inflammation, swelling, and pain. So some men did have highly symptomatic testicular inflammation after their injection.
Right, and that I haven't read that in the normal lists of side effects, but that's yeah. concerning. And Dr. Ros Jones, of course, has been doing some work on fertility effects. But if there was an injection where you've got naked DNA, then that's much more likely to give you a severe allergic reaction, Claire, is it?
Oh yeah, it is, and that's one of the causes potentially for the anaphylaxis.
Right.
And then also the technology involved large vats of E. coli being used to produce the DNA template, and endotoxin, which is this bacterial cell wall from the E. coli.
Yeah.
Is something that's really quite dangerous to inject, even at teeny tiny levels. It's almost not a dose response. Even the smallest level of endotoxin can cause problems, and the problems that most people are familiar with are dramatic. Anaphylaxis is dramatic. You don't miss that. It's happened, but there's very good evidence that it causes many more inflammatory problems, including chronic problems, which are harder to measure and easier to miss and easier to not associate because of the timing.
Yeah. And of course, it's endotoxins that give rise to a lot of the features of sepsis, life-threatening condition. Yes, endotoxins are, our bodies react very badly to endotoxins. They are quite dangerous at scale. And we're not talking about a sort of sepsis level of exposure here. This is a very small amount. But even that small amount can be problematic. Yeah, given the choice, I won't have any.
No, and the thing is that there are, that kind of manufacturing of a vat of E. coli at scale has many kind of economic advantages. And it means that you can do things like, what they do is they put an antibiotic resistant gene into the bacteria that they're using, and they can use that antibiotic and kill off anything else and just select out the ones they want. So that technology is great for them. And it used to be used to make insulin. That was how it was originally made. Okay. And so diabetics are at risk of having that endotoxin problem. And that's all been moved over to using yeast instead, where there are other issues, but there's not that endotoxin problem. And using yeast is slightly more expensive, and it has other advantages and disadvantages. But I just do find that interesting that that it was rejected for insulin production.
Very interesting. Yeah. Now I think what we're seeing here is an illuminated lipid nanoparticle that's been labelled so we can see it. Yeah, Luciferase.
Luciferase. Yeah, I was going to say they've chosen the right colour, because Luciferase is the gene that was used so that the lipid nanoparticles fluoresce under that immunofluorescent microscope. So when you see the nucleus, when I was saying you can see it got there, it's that colour on the when you get the images.
And of course, this is a bit of gene technology, because this luciferase is actually present in certain things that glow in the dark, isn't it? Like deep sea fish and various insects that I can't remember the name of. But
it is, of course, named after Lucifer, the light carrier.
Yes. We'll pass on from that one. You might.
Yeah, I think we should.
There might be some negative connotations in that. Yes.
I mean, it does get used a lot in science as a marker because of that ability to visualise it afterwards.
I mean, it is a useful scientific tool, isn't it? It lights things up and you can follow it. So it's very good.
Right.
Yeah. So we can follow those lit up nanoparticles as they are systemically distributed around all of the body. And when we say all of the body, we mean all 5 toes, your ears, and every single organ in every part of the body. It goes absolutely everywhere. And potentially with the lipid nanoparticles there as well. So this next video, Claire, we're looking at the modified RNA here. So this is not a natural physiological ribonucleic acid, is it? It's been jiggled around with.
That's right. So natural, our own mRNA, which is messenger RNA, that's what the M stands for, because it takes a message from the DNA to make it into protein. and it lasts half an hour, maybe an hour or two, and then it's gone. So it's I liken it to being I say if you imagine you're going to do some cooking in the kitchen, so you're making your proteins, and that you go to the computer database to get a recipe. So that's your DNA, and then you print it off on the printer, and that's your RNA. And then, by the time you've finished cooking, or maybe I'm a messy cook, but the paper's kind of rubbish and you throw it away. So that's like an analogy that I like to use to describe how it normally works. But the M here is for modified RNA.
Yeah.
So, or at least it can be for modified. It's sort of, being used interchangeably in a slightly kind of dubious way. So there was one company called CureVac who used sort of pretty regular RNA with the same lipid nanoparticles that Pfizer and Moderna were using. They did not get the same results. And there's various reasons why that might be, which are not all to do with the product they were using. But it was all attributed to that. So that what Pfizer and Moderna did is they took out one of the nucleotides. So you've got the 4 nucleotides that make up the letters that make up the sequence.
Yep.
They took one of those out and substituted it with this 1-methyl-pseudouridine nucleotide.
And pseudo, of course, means false. It's not the real uracil. It's not the real uridine. It's a false one.
And it's something that we don't have. We do have a tiny amount of it normally, but it's not something we have very much of at all. And here suddenly, all of the every 4th letter almost is this strange chemical. And what that strange chemical does is it stops the RNA being destroyed. So it sustains it for a longer time in the cell. So it sort of acts like a longer dosage. So you get more protein produced from it. And that there was a time when I was concerned that after this mRNA was broken back down into nucleotides, that there would be a situation where we had in our cells too much of this abnormal one, and it would be getting used in other protein manufacturing.
Right.
And that those mRNAs would hang around for too long, and the proteins produced would be too much, and it could cause all sorts of problems. But I was wrong on that. So the pseudouridine, the body doesn't use it, just rejects it.
That's reassuring. But it's good that you erred on the side of caution there, unlike other groups that could be mentioned who perhaps have not erred on the side of caution.
I didn't publish what I thought about that either. I just was thinking about it.
No, but it makes perfect sense that you could end up with this naked pseudouridine that could be a building block for future RNA molecules potentially, but thankfully that hasn't transpired to be a real problem. So here we see the RNA code, and in the background all these letters are the A, U, C and G. The adenine, it should be uracil, but it's pseudouridine, cytosine and guanine. And here we see all these letters lit up that are a synthetic base in the RNA. David Speaker actually described this as making it plasticky, that it hangs around for a long time.
I think it's quite a good description, yeah.
Now, we've got these pseudouridines here in the sequence. And basically, any 3 of these bases in this sequence will code for one protein. It's called,
1 amino acid.
Yeah, sorry, 1 amino acid, yeah. 1 amino acid, which is a component of a protein. So 3 code for an amino acid. Now, the idea is that if there's a pseudouridine, the body's, the base reading apparatus in the ribosome might not recognize this and might skip one. Is there any basis to this frame shifting idea, do you think, Claire?
Oh yes, absolutely. But all of that evidence came out after everyone had been injected with it. So although actually that's not entirely true. No, I think there was knowledge about it beforehand. and the evidence in this specific scenario about what exactly the frame shifting might look like was published after the event. And the publication showed that 30% of people who had been injected had antibodies to one particular protein that you might see if the mechanism slipped when it was reading the letters, which is what the frame shifting means. So it's like your hands have moved on the keyboard and you're typing a totally different word.
Yeah.
And but the thing is that was just one of the proteins. You know, there's any multitude of different things that could be produced, and or different lengths, or different, you know, all sorts of things can go wrong if you've got machinery that's just, you know, not aiming properly on a keyboard, which is what was happening.
So if you frame shifted, the three bases would read for the wrong amino acid. And you'd be putting the wrong amino acid in, therefore you'd end up with the wrong protein, and you could potentially end up with pathological protein production.
That's it. So you're just producing gobbledygook. And we know it didn't happen entirely because, you know, people did create antibodies to spike. So they were, you know, there was a kind of, there was the majority, or at least a significant amount, was doing what was intended, albeit whatever the intentions were, but it was making the spike protein. But there was other proteins being produced. There's undoubtedly other proteins being produced, and shortened spike proteins where the RNA had got fragmented, so you didn't have the whole story. It just cuts short.
Components of a spike protein rather than the whole thing. Yeah.
Right, and the thing is with proteins, their function is all about the shape that they're in. So the sequence of amino acids isn't really the thing. It's how they fold into each other to produce a shape that has a function. Yeah. And if you have a shortened one, that means that the interactions of them with each other when they're folding will be different, and you could produce something that's a very, very different shape and a very, very different function, even though it was nearly, you know, an entire spike protein. So there was all sorts of things being made for which we can't be certain what the function is. We don't even know what the sequence was, and consequently, you know, a lot of attention has been put on the pathology being related to spike, whereas in reality the pathology was so diverse. Yes. I think partly because it wasn't just spike.
Well, if 30% of people were developing antibodies to a foreign unknown protein, I mean, those antibodies would only be developed by the immune system if that protein was present. So the antibodies are actually a marker for the presence of this abnormal protein.
Yeah, which we still know nothing about.
Yeah. So I think what we're seeing here is this is the RNA, isn't it? Being read by the ribosome. And the ribosome is producing the this sort of wedge-shaped spike protein.
I like that graphic. I think you can see how that could stick onto the shell of a virus and look like a spike.
Yeah.
And there they go. And I think actually that image is quite useful as well, because although, like, in theory they wanted the spike to be in a cell that would get destroyed and therefore educate the immune system about destroying cells with spike.
Yeah.
But some of the spike got out of the cells and was circulating in the blood. And that was particularly bad with the AstraZeneca, but it did happen, I think, with the others too.
I mean, that's what you'd expect with production of foreign proteins, really, wouldn't you? You'd expect some to stick to the surface of the cell, but you would expect some to become humoral and to be distributed in the body tissues. Yeah,
and I guess that brings us back to this thing I was saying earlier about how things move in a cell. So there are these things called chaperone proteins whose job it is to transport so when a protein comes out of the ribosome, it gets taken to where it's needed. It gets taken to the cell surface, or taken to the nucleus, or taken to wherever it's needed for its job.
Yeah.
And these, maybe somebody does know, but I don't know what was going on there once this spike was produced. So you've produced a whole load of a foreign protein inside the cell, and some of it got to the membrane and stimulated an immune response. Some of it maybe got destroyed in the cell and then shown on the membrane to say, I've got this inside me, and am I for destruction or not? But I imagine some of it could just have been, well, if you've got something foreign, let's just spit it out, because that shouldn't be here. So the cells can, as well as sort of endocytosing, they can exocytose just whatever's in the cytoplasm that shouldn't be there.
Yeah, and again, exocytosis is a well-recognised sort of cytological process. This is nothing new. So we've got here, Erik is saying here about a 10% error rate. And I guess what we're seeing here is a pathological protein that's being produced as a result of this error.
Yeah, so I would put it at a high percentage. I think once you've included all the mechanisms for error, so the fragmentation of the RNA, the fact that the RNA sequence could be read backwards as well as forwards, which is really abnormal. Normally,
I never thought of that.
Normally if you try and read a sequence backwards, you hit stop codons again and again and again. So that, sorry, I've just jumped ahead, haven't I? So each of the 3 letters can translate into an amino acid, or it can translate into a thing called a stop codon, which stops the process. And there's quite a lot of 3-letter combinations that are stop codons. So normally when you turn a sequence back to front, you hit stop codons and nothing really happens. But with this particular sequence, there was this really odd absence of stop codons. So you've already got the kind of, well, you could do it the wrong way around error rate, and the fragmentation error rate, and then you've got the slippage error rates, and there's another one that I can't quite remember.
I had no idea these things could be read backwards. That's another sort of fairly concerning area.
Yeah, I mean, they shouldn't be, because each end of the sequence has got the sort of apparatus to say I'm the front and I'm the back.
Yeah.
But if it's fragmented, you'll be missing those ends.
Yeah. I mean, it's supposed to be, I think, just over 4,000 nucleotides in the messenger RNA that's given, but if a bit's chopped off, it would change the message.
Yeah.
Oh, here we see the activity in the nucleus, I guess, Claire. The reading of this is the nucleus. We've got the reading of DNA here. We can tell it's DNA because it's double-stranded.
Yeah.
So I guess this is the DNA reading enzyme or protein called polymerase II.
I think that's right. It's making the RNA.
Yeah.
So I think the point with this DNA contamination is, well, what was the DNA sequence of, and does that get made into RNA once that's in the cell?
Yes.
So, and obviously if it can be delivered into the nucleus, there's a risk that it ends up in that cell's DNA. And that does happen. It's not like that's just not complete scaremongering.
No.
Our cells do end up with bits of foreign DNA in them, and then our cells die, and it doesn't matter. But over history, our DNA does have evidence of bits of virus being incorporated into it.